welcome to the last Rough Cut seminar for the semester. We have our local girl talking today, so that's good. And before I get started, I just wanted to bring to your attention that International Mountain Day is next week, December 11th. I know you all knew that. But we're having a big event at the Emerson, and we'll be hearing about the snow science avalanche research that's going on at MSU, and hearing from people from the university as well as the Forest Service. So come on to the Emerson for the evening event, and hope to see you there. But today we have Laura Burkle. Laura came here in 2010, 11. She got her she did graduate work at Georgia and then at Duke, and she's a community. Is that right?
Dartmouth, the other D one.
Then she did a postdoc at Washington University in St. Louis. And she's a community ecologist. She's interested in plant and over to you. Thank you.
Thanks everybody for braving the cold out there. There are a lot of rosy cheeks and noses, so I appreciate it. The last one of the semester, so thanks for coming, and feel free to ask me any that occur between species across trophic levels, particularly the interactions that occur between plants and pollinators, especially bees. I'm going to try and show you some sunny, warm pictures to keep you all heated up instead of thinking about the cold outside. Historically, pollination ecologists have studied plant-pollinator interactions in isolation. So one plant and one pollinator, but we all know that these interactions occur in a much larger community context. And so this network approach can provide a really powerful representation of the ecological interactions that occur among species, highlights their global interdependence, and can give us a really good idea of which species and interactions might be particularly important for population and community dynamics. I made up this network here all by myself, but this is the general format that these interactions or these networks can be displayed. So often the plant species are along the bottom, the pollinator species are along the top. Lines connect plants and pollinators that interact, and the thickness of the line is proportional to the frequency of the interaction. And we can use these networks to look at their structure and their function to assess how different kinds of environmental changes might influence those components. And when I talk about function, it's particularly hard to assess function, but we're talking about pollination service. So about 90% of plants worldwide require insects or other animals for pollination. And pollination is essential for human well-being because we all like to eat food. So about 3/4 of the leading global crop plants worldwide rely on pollination. So I thought I'd start off by telling you about some of the big questions. They're really vague, big questions that I think about. And anybody looking at this screen? I can stand in front of it. Okay. So thinking about what determines species diversity and composition across spatial and temporal scales, and extending that to species interactions, what, in terms of especially the environmental conditions, what is driving their diversity and their structure and function. And so environmental conditions for me might be things like climate change, land use change, wildfire, invasive species, any kind of nutrient enrichment, things like that. And then how can we take this knowledge, most of it that I gain from sort of more pristine ecological systems, and use it to enhance pollination services in more managed systems like agricultural settings. So I thought I'd try and place these big questions in terms of research projects. This is the shotgun approach, which I've seen some others do, which may or may not be so successful, but I'm going to spend a little bit of time talking about each of these different projects, and hopefully if there's time, ending up with this one at the end. So these projects are in various stages of completeness. Some of them are nearing completion, some are ongoing, and others we've just started, and I'll be excited to get your feedback about those. So the first one was using both historic and contemporary data to investigate the effects of climate and land use change on plant-pollinator interactions over the last 120 years. And one new direction of this work is looking at more fine-scale mechanisms of CO2 enrichment and drought that might influence plant-pollinator interactions at a more local scale. The second project is extending some of these ideas related to species area relationships to species interactions. And so looking at how plant-pollinator interactions accumulate with increasing habitat area. Diversity and function. And then lastly, in a more applied vein, just a crop plants. So starting with this one here, as most of you likely know, forest communities in the US and elsewhere have experienced two major environmental changes in the past. Communities in the US and elsewhere have experienced two major environmental changes in the past century, loss of habitat and climate change. So habitat loss can have obvious negative consequences on the abundances of species and a bit more subtle effects on species interactions. Climate change or temperature changes can influence species phenologies, and interaction mismatches might occur if the phenologies of two interacting trophic levels get disrupted. So, for example, if pollinators can't learn to forage on and pollinate new plant species, that could have negative consequences in terms of mismatches for pollination service. Both of these photos were taken from our study site in Carlinville, Illinois, where most of the forested landscape has been converted to corn agriculture over the last century. But this study site, Carlinville, the town in Illinois, is unique because we also know a lot about how plants and pollinators interacted as well as their phenologies before any of these anthropogenic changes occurred. So in the late 1800s, this eccentric weird guy here, Charles Robertson, he meticulously documented all of the plant-pollinator interactions in the town of Carlinville. His study is still considered to be one of the most detailed, fully resolved plant-pollinator networks that exists. He was looking at 450 plant species, 1,500 pollinator species for over a total of about 15,000 unique species interactions. So he's doing this all in his horse and buggy, traveling around, documenting these interactions. In the meantime, a lot of land has been deforested, and there's been warming in Carlinville of about 2 degrees, primarily in the winter and the spring. In the early 1970s, John Marlin went to Carlinville to resample many of the same plant species, especially this one here, Claytonia virginica. It's also known as spring beauty. And what's cool is that he did this work as a young man. So he had just graduated from college, landed his first job, and was really excited to talk to us about his sites and his field methods, which was something that we weren't unfortunately able to do with Robertson. So that was really helpful to be able to interact with him. And then in 2009 and 10, we returned to many of the same study sites visited by Marlin and sampled the plant-pollinator interactions to many of the same plant species. And in addition, we visited any of the remaining forest out there. We had a lot of help in the field and in the lab. In particular, my collaborators on this work are Tiffany Knight and John Marlin. So thanks to them as I talk about this work here. So I thought I'd start off with a giant overwhelming figure. This is Robertson's original network of 26 spring blooming plants in the forest understory and the 109 bee species that he saw observing them. I have color-coded each of the 532 interactions according to changes that we saw in these interactions over time. So the black lines are those interactions that have persisted until today. So that's only about 25% of the original interactions that Robertson observed. All the other lines, the colored lines, are interactions that have been lost. And there's a bunch of different reasons why these interactions might be lost. In particular, due to species extirpation, so local extinctions of certain species might occur. In this modern fragmented landscape, there might be spatial loss of spatial co-occurrence of species that historically interacted. And there could also be changes in phenologies, abundances, behaviors, physiologies, things like that that might affect the propensity for a particular interaction to co-occur. So we're going to look at some of these possibilities, starting with extirpations of species. So none of the 26 flowering plants went locally extinct, so we were able to locate populations of all of them, so they didn't contribute to any loss of interactions. But we did only see 54 of the 109 bee species that Robertson observed. So about half of the bee community is no longer in this area. And of the 400 interactions that were lost, about 45% of them were due to bee losses. So the bees in red up the red lines are interactions that were lost for that reason. If we just look at a general breakdown of what happened to all these interactions, what contributed to their losses, this chunk here, these are the 25% of interactions that have been maintained. And this large chunk represent the interactions that were in red in the last figure, so those that were lost because of extirpations of bees. Because of loss of spatial co-occurrence due to habitat fragmentation or loss of temporal co-occurrence due to changes in phenologies, those together make up about a fifth of interaction losses. The big orange chunk are interactions that were lost for reasons other than we measured. And so these could be physiological shifts, behavioral shifts, shifts in abundance that have been together sort of deemed mutualism abandonment. So the species are still there, they're still active at the same time, so they could occur, but they just don't. What we found particularly concerning was when we took a closer look at these maintained interactions, and they're really tenuous. So they often occur only in one forest fragment or for a really short temporal window. So about 75% of these interactions occur or could occur for about a week or less. So the phenological overlap between them was 7 days or less. So further habitat loss or further phenological shifts could have pretty negative consequences for the remaining interactions. So all of these changes in network structure, shifts in bee diversity across the landscape could affect function, so it could affect pollination services. And so we wanted to try and look at that for one really common plant species, Claytonia virginica. It's really common in this area. It's important because it blooms at this beginning of the season when bees are just emerging from winter and looking for stuff to eat and pollen to provision their offspring with. So we were lucky in that we could look at this plant and its visitors for all three time periods, so Robertson's time period, Marlin, and currently. And so maybe not surprising to you at this point, we found that the diversity of bees visiting Claytonia has declined over time. What's kind of interesting is that Robertson and Marlin saw a very similar diversity of bees, so no declines in the bees visiting Claytonia between 1916 and 1971, but have declined by about half since that time, so in the last 40 years or so. And we were able to link this decline with changes in land use. So again, maybe not super surprising to you, but the loss of forested habitat, and in our case, at the same time, increases in development, primarily residences. Sites that lost a lot of forested area also lost a lot of bee species. So although it's intuitive, rarely do we actually get to document a lot of these patterns over time. To me, one of the most exciting parts of this project was our ability to look at the pollen on the bodies of bee specimens and use that pollen as an estimate of bee fidelity. So how promiscuous or not the bees are being as they move from flower to flower. Are they visiting all the same species, or are they shifting their behavior to visit flowers of different species? A high degree of fidelity would indicate that those bees are going to be pretty good pollinators, that they're going to be very efficient and effective in pollination service. So here's the Illinois Natural History Survey, which is where Robertson's collection as well as Marlin's bees are maintained. To wash the pollen off of their bodies. That's a 120-year-old bee from Robertson's collection. And things like that, but it all worked out. And so then we can take those slides and stain them and look at the pollen. And so high fidelity to Claytonia, low fidelity would be down at the bottom. So you can see that services. We did some studies looking at Claytonia reproduction currently across different forest fragments, but we don't have any data from Robertson's time, so that's where we ended up. So what we're doing now with this general topic is starting to look at some more fine-scale mechanisms of how climate change in particular might influence the way pollinators perceive plants. And this work is in collaboration with Justin Runyon. You want to say hi? So Justin works for the Forest Service, the Rocky Mountain Research Station. He's a chemical ecologist as well as an entomologist. And so we're using his expertise in volatiles to try and understand how floral volatiles, or floral smell, as another way of saying it, is important to pollinator attraction and how that might influence plant-pollinator interactions at the community scale. So we're doing this at Mount Ellis, which many of you might be familiar with, about a 15-minute drive away, Wildfire Meadow. And for the past couple summers, we've been documenting the general network of interactions there. So far, we have looked at visitors to about 50 different plant species, and there's over 70 different bee species in this community. And we also have evidence that the smell, or the volatiles that these flowers are making, influences which pollinators come visit those flowers. So how do we do that? Well, Justin has these amazing portable volatile collectors. So a vacuum basically pulls air across a flower and collects volatiles on a filter, which we can run on gas chromatography to get an idea of which compounds are present in the flowers of which species. And so far, we found this interesting relationship where each of these dots is a different plant species. And so the greater number of different compounds that are present in the smell, so a more complex floral bouquet, so to speak, gets visited by a lot more bee species, except for these three colored dots here, which are three really common flowering plants in this system. So Potentilla, Arnica, and Geranium. So we're interested to see sort of how these, if the abundances of these plants is really driving some of these patterns as well as their volatiles. So our next step here is to grow plants in growth chambers, different forb species under enriched CO2 in combination with drought. We would like to be able to manipulate temperature as well, but we don't have enough space in growth chambers, so we're going to focus on CO2 and drought for now, assess their volatiles, and bring those plants out into the field to see which bee species might visit them in comparison to control plants. One other thing that we'd also like to do is assess herbivory of these plants. So herbivory might be affected by the CO2 and drought treatments, as well as the fact that herbivory also alters directly floral volatiles. So with an N of 1, we have one past flower here that did not have thrips, so insects that eat pollen, and this one did have those insects in the flower. And so you can see that herbivory may induce volatiles of these different groups that might influence subsequent pollinator visitation. And then lastly, as a sort of extension of this work that we're doing with Kathy Zabinski and LRES is inoculating certain species of forbs with certain species of mycorrhizae and measuring floral traits to see if certain combinations of these below-ground interactors might affect above-ground mutualists. We'd like to take what we learned from this greenhouse study, incorporate it into our some of our growth chamber experiments, perhaps doing some mesocosms out in the field at some point to combine both below-ground and above-ground players. Okay, so that was sort of the bulk of a lot of projects that we're working on. I wanted to describe this interesting project, which is looking at how species interactions accumulate with habitat area. And so we did this work in Ozark glades in Missouri, glades that vary in habitat area, and looked at biological mechanisms that might drive some of these patterns that we saw. So as you might know, one of the oldest relationships in ecology is the fact that the more area you sample, the more species you get. You can view this as a saturating curve or as a straight line on a log-log scale. And there are a number of different reasons why you might see this kind of relationship. Habitat heterogeneity, low extinction rates in large populations, as well as passive sampling. And these relationships have been used in the past and currently to try and optimize the size of conservation areas. So you want to know how big of a habitat you need in order to conserve the greatest amount of biodiversity. And so we wanted to kind of extend these ideas to species interactions, because we would argue that the biodiversity of a place might not be able to be maintained unless you also have the interactions present that are required to keep those populations going over time. So in our case, seed set of plants related to pollination. So having all the interactions present that would also maintain those, the fitness and reproduction of those plant populations. There have only been 2 studies to our knowledge that took a look at this question. So how do species interactions differ in habitats of different sizes? And they both found the same thing. So the number of interactions per species, so links per species, number of interactions per habitat area. So another way of saying this is that interaction richness increases faster than species richness does in larger habitats. But this pattern of interaction richness increasing with habitat area could just be a passive sampling effect, just based on the fact that bigger areas have more individuals and more individuals have more species. And so we wanted to test whether or not this expectation was just due to chance or if something else was going on. So if we saw observed patterns that were either steeper or shallower than the expected, something interesting biologically might be going on. So 2 biological mechanisms that we looked at, not mutually exclusive, are the overall diet generalization of species might vary with habitat area. So here we're talking about species composition. Habitat areas that are larger might have more generalist species or more specialist species, which could lead to deviations from this null expectation. Of a particular species might change their behavior and change their diet depending on what else is in a habitat. And so we could get this same steeper than expected pattern if individuals are being less selective in their foraging in larger habitats. Perhaps there's less competition for selective in their foraging. So they're able to focus on their preferred food. If you really want to eat Twinkies and there's no competition for them, maybe you will focus on eating them. So we were interested in asking this question of how and why do species interactions in habitat islands that vary in size. And we use these Ozark glades as our study system. So if you've never been to one, a glade is sort of an open, grassy, herbaceous area within a forested matrix. And historically, these glades were present over really large areas of limestone bedrock, but because of fire suppression and trees invading, they've become much smaller in size. So we were able to select glades that ranged in area from maybe twice the size of this room to about a dozen foot. And what we found was that occurred more quickly than species richness did with increasing habitat area, in this case measured by the number of individuals. However, when we modify the number of individuals, However, when we modeled the expected number of interactions across the same range of pollinator individuals, the observed was much less. It was shallower than expected. So this is on a log-log scale, so these differences are bigger if you show them without that. So this pattern of having a shallower than expected slope could result from having more specialist species in larger glades or more selective foraging. So we wanted to look at those two. So for the first mechanism related to species composition, you could think of that as the diet breadth of pollinators that occurred in these different glades. They became more specialized, so more specialist species were present in larger glades. So this would concur with this expectation. So a shallower than expected slope resulting from shifts in species composition. The other mechanism related to differences in foraging more generalist or less selective foraging in larger glades. And so if you remember, this pattern would have resulted in a steeper than null expected slope of this line. So that was going in the opposite direction of what we actually observed. Just to summarize these sort of mathy results here, we saw a shallower accumulation of interactions than might be expected just by sampling. And this resulted largely from shifts in species composition to there being more specialist species present in larger habitats. And because habitats of shifts in species composition would have to be particularly strong to outweigh those. Those other effects. So I just wanted to point out one of the implications of this work, bring us back to something a little bit more real. Because of the fact that these interactions accumulate more slowly with habitat area than we expect. So larger habitats might be needed to conserve those interactions than we might think based on species area relationships. Okay. This third project I'm very excited about because we've just started it, and it's a really fun collaboration looking at wildfire and productivity as drivers of plant and pollinator diversity and function. I'm not a fire ecologist, and so I'm learning a lot of new things as we go. As you guys probably know, wildfire is really important for ecological systems, maintaining diversity and sort of driving patterns of species composition across space. We also know that wildfire disturbances, their regimes have been disrupted or changed over time and are predicted to become more intense and over larger spatial scales than they have before. There's sort of this dueling use of resources where there's billions of dollars used to suppress and control wildfire in order to protect human communities embedded in fire-prone landscapes, as well as spending huge federal investments to restore fire's role in the landscape without a good understanding of how many different kinds of species and their interactions might be affected. And so given my interest in plant-pollinator interactions, you might expect that this is sort of the focus of where we're going in terms of looking at how these complex networks of species interactions recover from wildfire disturbances. Maybe I shouldn't say recover, but they are altered by wildfire disturbances at the scales that are spatial and temporal scales that are particularly relevant to conservation and management. And so one of the ways to do this is to look at site-to-site variation in species composition, so beta diversity, and site-to-site variation in species interactions, which we're calling interaction beta diversity, across large regional gradients like productivity. So these are just a couple pictures of some of our field sites. And so taking a closer look at productivity, variation in net primary productivity is hypothesized to strongly mediate how species might respond to disturbances. Hopefully you recognize this as a map of Montana. Brown is low productivity, all the way up to this blue-green color is high productivity. So we've selected some sites along this gradient. And you might imagine that highly productive areas might recover quite quickly from disturbance. Each site within a highly productive area might recover differently because there's a large regional species pool, which could lead to high beta diversity across the landscape, whereas low productivity areas might respond more slowly, might all respond similarly in terms of species composition, so having lower beta diversity.
These are all forest sites?
Forest sites.
All lodgepole?
Not all of them. Yeah, so species composition varies quite readily across there. To potential historically to have been many mixed severity fires across the landscape, which might lead to high beta diversity because of this sort of mosaic structure of forests, differing environmental conditions and habitats across and timber harvesting, along with some degree of climate change, might cause fires to burn with higher intensity and over larger spatial scales, which could homogenize forests and environmental conditions and could lead to low beta diversity across the landscape. So sort of in this potential context, we wanted to test these ideas using a wildfire chronosequence, so over about 30 years of fire data across an NP peak gradient. We decided to use this chronosequence approach because we could look at fires at large spatial scales. Nobody's going to let us burn a really big area, and so using sort of older and newer fires, we can get a more realistic view of what's going on. So currently we are comparing old wildfires, so 25 to 30-year-old fires, to more recent 5 to 10-year-old fires. Ultimately, we'd like to have super new fires, year 1, year 0. We're working on that. And we're doing this across this productivity gradient. So Helena has the lowest productivity, Whitefish has the highest. And we're also comparing different kinds of fire types. So areas, this is say a 15-hectare area that has a large mixed severity fire. So high intensity spots, medium intensity spots, low intensity spots, all within that area, compared to blocks of high severity fire of the same habitat size. And I'd argue that plants and pollinators are a particularly good system to address some of these questions because pollination is needed in the recovery process, and because we know a lot about what happens to the plant community, both above and below ground, after fire. So when canopies open up, lots of wildflowers come in, but we know very little about how fire directly affects highly mobile pollinator communities. A lot of these guys nest in shallow earth or in twigs and stems, which obviously could be burned up by a big fire. And as well, pollinators might be influenced by these resource pulses or shifts in floral resources through fire. So direct and indirect effects, fire could influence, or the spatial scale and the temporal scale at which fires occur could influence plant and pollinator communities as well as their interactions. So we've just started this project. Please give us lots of feedback. We've been establishing 180 transects across these 3 different levels of productivity, looking at both old and newer fires, as well as comparing mixed severity fires to high severity fires and unburned locations. The pollinator and plant-pollinator interactions along each of these sites. We've been quite busy identifying things and still processing a lot of these data. So I'm going to show you a few slides of results and just say that they are very preliminary. Come back to that. So the first thing we wanted to look at was just a tally of the number of different plant species along each of these transects, break it up by locality, so productivity, low to high, and break it up by the different fire types, unburned, mixed severity, and high severity. The white numbers are the number of replicates that we've processed so far. So Helena has been completed a lot more than, say, these other ones over here. Despite that, we're looking at new fires, and you can see that there's a trend in the direction that we expected for there to be more species with higher productivity. We also see greater richness of plant species in mixed severity fires compared to the other two treatments. And there's this interesting productivity by fire type interaction where high severity fire has the most negative interaction on species richness only in the most productive sites. So if we look at these same data but look at species composition in ordination space, so each of these dots is a transect. Dots that are closer together have more similar species composition than those that are away from each other. You can see right off the bat that Helena has quite different species composition compared to the other 2 sites. You can also see that within a locality, the fire types cluster together. So high severity fire clusters, as does mixed severity and unburned within a locality. What we were really interested in looking at was the beta diversity, so the dispersion of these different fire types. So spots that cluster closer together have low beta diversity compared to those that are spread further apart. If we just look at Helena, because we have the most data so far, you can see that high severity fires have the lowest beta diversity, mixed severity fire intermediate, and unburned has the greatest beta diversity. So we'll be excited to see what happens when we process more of those transects in paradise and whitefish, if we see more similar or different patterns. If we just look at Helena, at the high severity fires there, and compare fire age, so new, old, and unburned, the older fires are supporting greater plant diversity compared to the others. So this makes sense perhaps in a successional context where older fires might have some species characteristic of newly burned areas as well as some species characteristic of unburned spots. Again, if we look at species compositional data, we see that that sort of trend where old is intermediate between new and unburned areas. As well, beta diversity seems to increase or develop across time. So lowest beta diversity in the new fires, larger with old, and greater with unburned. We're really excited to see if the magnitude of this effect differs at other locations. So Helena is really low productivity, but if we look at, for example, whitefish, whether or not we'd see similar or different results there as well. So we'd love to be able to link all of these patterns that we're seeing in plants and hopefully in pollinators soon, as well as plant-pollinator interactions, linking those patterns to actual effects on pollination service. And one of the ways that we can measure pollination service is to pick one or a couple plant species that occur across all of our localities and do an experiment where we compare hand-pollinated plants. So we pretend to be the bees, and we go around and we give plants maximal pollination and compare the fruit and seed set of those plants to naturally or open pollinated plants. So the hand-pollinated plants tell us how many seeds and fruits that plant can make given the environmental quality of the area, soil nutrients, sunlight, whatnot. And the naturally pollinated plants tell us how many seeds and fruits are created given the pollinator community and those environmental conditions. So the difference between the two tells us how pollen limited plants are and how well those pollinators are providing service in a particular area. The problem is that it's really hard to do this kind of experiment, and so I'm curious to hear your feedback on whether or not it's worth it. Perhaps it's been something that we've been talking about a lot, and whether or not we should try and actually do Objective 3 of that proposal that we wrote. So anyway, I will leave it at that for this project and just say this has been a really fun summer. We had a lot of help from our wonderful field crew. This work is in collaboration with Travis, who's here in the front. Travis works at the Wilderness Society in town, and Jonathan Myers, who's at Washington University in St. Louis. I am going to, what do you guys think? So there's 10 minutes left. I'd love to hear your questions. So I'm just going to say, for this part of this project here, this is another new project that we just started this summer with Casey Delphia, and CC Welsh was also involved. We planting perennial flowering strips on CSA farms, and with the idea that the proximity to these flowering strips, the proximity of crops to these flowering strips might help their pollination. So pollinators being attracted to these plants once they flower, and then going on to visit other crop plants. So we planted some test strips of squash and sunflower to assess how these plants might influence pollinators. So with that, I will say I'm done. Ted.
So, I mean, you've given us so much to ask.
So if you guys could have these mics and have a few.
Be recorded for all time now. You're recorded for all time now in your question. Right, now I've forgotten that. So you were talking about the species area effect.
Yeah.
So I think that the area available for preserves is fixed.
Okay.
Okay. So how are you going to decide how to divide it up would you rather have one big one, or would you rather have a bunch of tiny ones, or what's the optimum in there you're wanting to optimize?
I believe this has been debated for all time, so I'm not going to be able to drive to. And so where does it saturate? You know, is it linear, is it nonlinear? And say, okay, we get the most species and the most interactions at this point, and then above this point, it's all the same, perhaps. And so then you could divide up an urban garden, you would definitely get more species that aren't present in those glades. But if you're just interested in the glade community,
accumulating, you're not getting as many interactions as you might, almost as if they're missing interactions. Accumulating, you're not getting as many interactions as you might, almost as if they're missing interactions. Couldn't that just be the way interactions accumulate? You're getting more specialist species. That idea that maybe you have to go even bigger to get the interactions. But I was
service. And so for that project, we didn't see any differences in fidelity across service. And so for that project, we didn't see any differences in fidelity across habitats of different sizes. So you're right. That was possible that more interactions isn't better.
or they just aren't there.
Or they just
like you'll get another you add area and you'll get another specialist.
The curve. Yeah, and so it seems like that's playing a big role. But then there's also these more generalist. They're foraging on more things in bigger areas, which we found interesting. But they weren't sort of specializing. But apparently, it doesn't matter for their pollination service. Yeah, I think data. The fact that they were abundant was more of a afterthought, like, oh, who are those dots up there? Who's messing up our relationship? Afterthought, like, oh, who are those dots up there? Who's messing up our relationship? And oh, it's those three. They're really important. They're like the most abundant in terms of flowers. Yeah, but yes, that's a great idea.
I was surprised in the big Illinois study that
you had a big drop in bees, but there wasn't any drop in plant species. So there's a lot of redundancy going on, or what?
Yeah,
I mean, I
so,
couldn't tell from the big graph what the explanation for that was.
well, I mean, we don't know for sure, but
But I mean, it's a
plants
huge
are,
study over so many years. And,
yeah, we don't have any estimates of abundances, particularly of plants or flowers from the late 1800s. He didn't record that sort of information, unfortunately. But what we do know is that the plants are perennial and that the bees are basically annuals. And so plants can likely handle a year or 2 or 3 or whatever without reproducing, whereas the pollinators might be especially sensitive to fluctuations in plant resources or nesting habitat or whatever it happens to be. And so they might weak out of some of these fragments and have a lot of trouble recolonizing them, where plants could hang on for longer and figure out how to deal with issues. Yeah.
So how does that relate to the Claytonia pollen on the bees? And so there's a lot of Claytonia on the initial study, and then the last first of all, what other plants were they visiting? Were they native?
They were native. So we focused on the spring understory forest community, and most of the invasive species were present sort of on roadsides and later in the summer. So we didn't see a whole lot of non-native plants or their pollen showing up. So all the other pollen was typically native, and it's often what is common in that forest fragment that's left over. Does that make
It
sense?
shows that pollinators are also quite generalist in what they can.
Some of them are,
Seriously impacted
by the loss of plant species. I think it depends, because I didn't show this either, but most of the bees that were lost were specialists. And so perhaps their one plant that they do visit is not present in the forest fragment, and it's the generalists that can, deal with changes in floral resources that are present there. So the species of, we chose 6 species of bees to look at the pollen on their bodies for that study, because we had access to enough of them, which usually means that they're common and generalist in their foraging. Although we did have a couple of kind of specialist species that we were able to access. We're not allowed to use the holotype specimens for any of this kind of work. So it's possible that we were looking at those bees that were more flexible in their diets to start with.
I think there was a question. Did everyone see a question flash on the screen from Rick Soja? Do you know that, Nadine?
It flashed on the screen, right?
Are none of the pollinators migratory? Even if locally, how does this affect any assumptions about escape?
Okay, so migratory. Unfortunately, bees don't fly very far, typically. 300 meters from their nest is the general consensus for a lot of solitary bees. Bumblebees and honeybees, well, we're not going to talk about honeybees, but bumblebees can fly a bit further. So migratory isn't a word I would usually use to describe bees. And so if they, go locally extinct in one of their forest fragments but can't get to another
were there any interesting? Did you identify these volatiles that came up with the herbivory? There are lots of new volatiles.
of, I have to go way back.
Sorry about that.
Sort of typical of herbivory and the kinds of compounds that are induced in leaves when herbivory occurs. So the 4 different groups are common, but I'm not. You can ask.
Herbivory in most plants that have been looked at. So those are, that's a well-known phenomenon. Green leafy volatiles are released passively when green tissue is damaged and cells are broken open. But as far as all the compounds are known to be emitted by other plants. It's just interesting that feeding changed the floral volatiles. Most studies have looked at the leaves.
I see.
This